The human brain is the most complex structure in the known universe. This is an exaggeration, just when one considers the incredible number of synapses, its plasticity. There is no other structure which has been seen through a telescope or a microscope more complex than the human brain. This video is going to start to go through some of its sections and then relate that to other vertebrates and talk about the vertebrate brain. Now, because the brain is so complex, it obviously makes sense to break it into parts. And if one were then to look at the human brain, we see 6 regions. The largest region is the cerebrum, which is greatly expanded in mammals, even more so in primates, even more so in higher primates and humans. But that's not where I'd like to begin. The second largest part in the occipital region of the skull is the cerebellum. That also will be covered later, as will what we see in the center here in green, known as the diencephalon. What I'd like to start with is these 3 structures here, which are collectively called the brainstem. So when the spinal cord enters the foramen magnum and expands to become the brain, it starts to form this region here, which contains a red region known as the medulla, a blue region here known as the pons, and then this orange region known as the midbrain. Once again, collectively, they are called the brainstem. And I'd like to just quickly mention a few topics related to the brainstem and that of the vertebrates. Now, most of the brainstem is made of white matter. So when one looks at, say, a cross-section of the brain, you can see that there is a greater concentration of soma, which make the cortex of, say, the cerebrum or the cerebellum look gray by comparison. When one looks at a cross-section of the brainstem, it looks white. Now, white matter contains more axons because the axons are often coated with the lipid protein mix known as myelin, which gives them a lighter color. And if the largest 2 parts of the brain are the cerebrum and the cerebellum, it obviously then makes sense that the brainstem is mostly white matter because we need fibers which are going from the body to these largest regions of the brain. They are obviously ascending tracts. And then these large parts of the brain then communicate with the body in descending tracts. So the cerebellum sends fibers to the body, the cerebrum sends fibers to the body, and then obviously then these large areas have to talk to each other as well. Although most of the brainstem is composed of white matter, I have these little outlines in gray here where there are nuclei. The term that we use for a cluster of soma when it is inside the central nervous system is a nucleus, or in plural, nuclei. And so while the brainstem is primarily white matter, transmitting messages to and from the larger parts of the brain and the body, nevertheless, there are then also sets of gray matter or nuclei. Now, these can be referred to individually by names like the red nucleus, the nucleus of the ocular motor nerve, etc., or they can be collectively called the reticular formation. So just in general, if we were referring to all of these nuclei that start in the medulla and the pons, in the midbrain, there's even a nucleus of the reticular formation in the thalamus, which is in the diencephalon in green. Sorry about that. So the reticular formation then includes pathways of axons, which are going from the body up to these large parts of the brain. They're carrying sensory information. And collectively, these nuclei then help us to focus attention because the information that we get from our extremities too much. I can feel my shirt here. I can feel the chair. I can feel touch here. There's pressure, there's pain. But obviously not all of it is important. I'm going to focus my attention on a small amount of that. The reticular formation helps us do that, to sort out those stimuli coming in which aren't urgent and don't need attention. How alert we are and where we are as far as our sleep awake cycle. Are we drowsy, etc.? These help to determine that. There's also a lot of movement, which is determined here. Now, it's very easy not to give importance to the reticular formation and these nuclei because we're not aware of them, right? So I'm aware of the thing that I'm focusing attention on, like something in my hand, or I'm aware of this movement that I just planned and that's going on in the cerebrum. Isn't the cerebrum then the most important? Well, it certainly is, but these involuntary functions, which are being carried out by the reticular formation, they're important. Decisions are made about my breathing. Should I be breathing more or less? Decisions are made about my heart rate. Should my heart rate go up or down? Should I blink if there's a loud sound? Should I turn my to my head? Which of these stimuli are important? I'm not aware of the processing that's going on in these nuclei. But obviously that doesn't mean it's not important. So the reticular formation, all of this is involuntary. We aren't aware of any of it. But nevertheless, it's a very important level of processing. When we look at the reticular formation in other vertebrates, we can see that the simplest vertebrates, the jawless vertebrates, they had some nuclei in their medulla, their midbrain, even the thalamic reticular nuclei, but they're simpler than what we would find in the jawed vertebrates, the mammals, etc. I just like to make a quick point. You may notice that there is no pons. So the pons as an anatomical structure is not recognized outside the mammals. However, the nuclei which mammals have inside their pons do exist in animals which lack a pons. So even though you can't recognize the bulge of the pons, the pontine nuclei do exist in more primitive vertebrates. As you then compare, say, the reticular formation of a jawless fish to that of, say, a shark, you're aware that it's similar in its structure, but the clusters of soma, the nuclei from the jawless fish, there's now more neurons, and they are more subdivided than what we would find in the jawed vertebrates. So there is certainly homology. It's not as if these are two completely different structures, but as you start to look at the shark, as you see here, there's a greater number of nuclei which were smaller and more generalized in the jawless fish. As you compare the reticular formation of, say, the jawless fish the sharks to the amphibians, the bony fish, the reptiles, they are largely similar. There are, some changes which, appear in the bony vertebrates or in the amphibians, particularly now that there's arms and legs whose movements have to be coordinated. But in general, nothing overly dramatic. As we make shifts into then the mammals, you can see here there are some changes which occur, but for the most part, this reticular formation is largely homologous then as we go through the different vertebrate groups. Now, the reticular formation is the collective name for all of these nuclei. But then we could also then name them individually and speak of specifically the ones, say, in the medulla. So the medulla is, or the medulla oblongata, this is the first region of the brain after entering the spinal cord. There are descending tracts that go through it. So the pyramidal system of the commands that go to voluntary muscle get its name from the bulges on the medulla, which are called pyramids. They roughly look triangular. So obviously the corticospinal tract and the voluntary command over muscle goes through here. The ascending pathways must go through here. So in the posterior column transferring touch information, there's a bunch from the leg and hip called the fasciculus gracilis, and a bunch from the arm and shoulder called the fasciculus cuneatus. They are destined for nuclei in the medulla, the nucleus cuneatus and the nucleus gracilis. There's the major cardiovascular center, which is determining heart rate, blood pressure, blood vessel diameter. There's the major breathing centers, which will then determine the breathing, the rhythm of breathing, including autorhythmic fibers, which will fire spontaneously. And then some of the cranial nerves, which leave the brain and then go to the head region, have nuclei here, such as the cranial nerve 8, which will go and innervate the senses of the ear, cranial nerve 10, the vagus nerve, which is a very big nerve for the parasympathetic division of the autonomic nervous system, and others. And so the medulla, this is the first region. Let me talk about the pons and then mention more primitive vertebrates for a second. Here you can see this very prominent bulge that we have as the pons. And the reason for its prominence is if you look where it's situated, it is at the intersection of the two largest parts of the brain. So the cerebrum and the cerebellum mix here. And so obviously this is where there's a traffic jam, to use an analogy. So this is where fibers are making a turn to go to the cerebellum, or fibers from the cerebellum make a turn to go to the cerebrum, or fibers from the cerebrum are making a turn to go to the cerebellum. And when we look at other animals, we'll look at other animals in a second. So because so much is being sorted out here, there's a lot of axons which go back and forth. Now, there are some nuclei as well. So there are some areas which contribute to breathing, such as marking the transition between inhales and exhales, or doing the transition between inhaling and exhaling. And also then some cranial nerves have their origins here. So the pons is not just, it isn't just axons. It is, there are some nuclei as well. Most vertebrates don't have a pons. No fish has a pons. So you'll see that's missing in these regions of the brain. No amphibian or reptile has a pons. We simply progress from the medulla into the midbrain. If you look here at the opossum, you can see, oh, look, there's a little bulge. There is a little pons. What's going on there? Why do mammals have a pons? Well, mammals start to use their cerebrum more. A lot of what is primarily run by the midbrain in reptiles then starts to go into the mammals. The mammals get far more neurons in their cerebral cortex. And so, and in addition, in mammals, the cerebellum then becomes more developed. And so as you're seeing more neurons in the cerebellum and the cerebrum, then you get more development in the pons, which connects the two. Here in a cat, you can see the pons is prominent. Here in the sheep, you can see the pons is prominent. Once again, you notice that the brainstem is mostly white matter compared to the gray matter in the cerebellar cortex or the cerebral cortex. But we're starting to get more of a bulge for a pons here because of the cerebrum and cerebellum there. Primates grow the cerebrum and cerebellum even more, apes even more, humans even more. And so when you look at the pons in humans, it's extremely prominent. But once again, that's a testament to the great expansion of both the cerebrum and cerebellum, that here this area where the the fibers get sorted out, must then expand as well. Let me just kind of take a step back and then talk about the
primitive chordates have a much smaller brain region, although it does contain a medulla-like posterior region.
So going to some of the evolution, in chordates, a lancelet doesn't have much of a brain. The part of the brain that it does have seems to be medulla-like. And so this arguably seems to have, roots farther back in chordate history than most regions of the brain. The jawless fish have some advances in the medulla, such as they have ventricles, which enclose cerebrospinal fluid, which was lacking in the chordates. And the medulla is actually the best developed region of, say, a hagfish or a lamprey brain, and could make up half of its length. We certainly see similarities between the medulla here, such as the modified capillary beds where cerebrospinal fluid is made, the nuclei of cranial nerves, which we see in the higher vertebrates. Once again, as we start getting into the medulla of the jawed vertebrates, including sharks, as you can see here, there are more advances. The same thing here with the amniotes. So here you can see there's a medulla in the turtle, the alligator, the bird. So this is a constant region of the vertebrate brain, and once again, arguably may even go back to more primitive chordates. And at each step, if we wanted to get into great detail about the nuclei, the tracts, et cetera, this is perhaps more detail than most students are interested in. You could see things which the jawed vertebrates share, and then the tetrapods share, and then the amniotes share, and then ultimately then the mammals share or primates share. And so the medulla is certainly homologous. Once again, there are things which correspond, throughout the vertebrate groups, but then it clearly has evolved over time as well. And the same thing with the pons. There are, regions that we mammals associate with the pons present in vertebrates which lack the pons, but we don't actually get to a pons until you get to that the large vertebrate brains. In this last section of the animation, it starts off with essentially the brain of a chimpanzee. And then, so here's the brain of a chimpanzee. Now it has a pons here in blue, and a chimpanzee does have a big cerebrum and a cerebellum. But as we go from a chimpanzee-like brain to a human brain, there is a 3 times relative expanse, not just of the cerebrum, but of the cerebellum as well, which is why we have such a large pons. The last region of the brainstem is known as the midbrain. Now, as human embryos develop, I'm sorry, I'm having trouble pausing. So I'm going to try this other question set. I apologize for these temporary glitches. So if we then consider the midbrain is the smallest section of the human brain of the 6 regions, and therefore it's easy to disregard, perhaps once again because it's not the biggest. Nevertheless, it does have important tracts going back and forth, obviously. So the corticospinal tract giving voluntary information, commands to skeletal muscle must go through the midbrain to get to the muscles. The information about touch are going through the medial lemniscus, traveling here. And there are important areas where there are soma, such as the superior and inferior colliculi that I'll mention, the substantia nigra, which controls muscle movements, and the nuclei of some nerves. So the midbrain is extremely important in other vertebrate groups, and so perhaps worth a little bit more attention here. Of the three parts of the embryonic brain, the prosencephalon, the mesencephalon, and the rhombencephalon, all of the other sections become subdivided except for the mesencephalon. This becomes the midbrain and the midbrain only here in orange. To see the midbrain well, you obviously have to remove the cerebrum and cerebellum, and here you can see it here. Another easy way to identify where, oh, and I'm sorry, so obviously here you have the midbrain. Notice that in so many of the other vertebrates, there's what's known as midbrain exposure. So if you look at a brain, you say, oh, there's the midbrain right there, there's the midbrain right there. But obviously in most mammals, and certainly humans, the incredible growth of the cerebrum now has covered the midbrain and ended that midbrain exposure. So it is no longer exposed on the dorsal surface of the brain that way it once was. An easy way to find the midbrain is to think about the spaces where cerebrospinal fluid passes. Cerebrospinal fluid is made in ventricles, goes from lateral ventricles to a third ventricle. Here you see a pig brain developing. There's the lateral ventricle. But from there, it goes through a channel known as the cerebral aqueduct. And once you have found the cerebral aqueduct, you then have found the midbrain, because the midbrain, the cerebral aqueduct goes through the midbrain. And so once again, if you're trying to find this smallest region of the brain, finding the cerebral aqueduct is important. There's the cerebral aqueduct in the sheep. Sorry, let me just go a little slower. So once you find the cerebral aqueduct, you say, oh, here's the midbrain. And as we'll see, these are those bumps. The superior colliculi where vision is processed, the inferior colliculi where hearing is processed. The cerebral aqueduct leads into the fourth ventricle, which is at the base of the cerebellum. Okay, and so if you were to look at a certain section of the brain and you would say, oh, there's the cerebral aqueduct, then you would realize, oh, I'm looking at a section of the brain which includes the midbrain. So the cerebral aqueduct splits the midbrain into two parts. The tectum is the part that is over it, and that then can be divided into the superior colliculi and the inferior colliculi, while the part of the midbrain which is beneath the cerebral aqueduct is the cerebral peduncles, which we could then subdivide as well. Once again, we could talk about all of the subdivisions, and I do get into this a little more in these next videos, so why don't I do that? We could talk about the nuclei, which are here. Once again, they are all involuntary, so it's easy to overlook them. But so many of the reflexes that they give are quite important. And so, sorry. So one of the nuclei in the reticular formation is the red nucleus. And the tract is called the rubrospinal tract, which goes down from and out to muscles for muscle control. The ocular motor nerve has its nucleus here in the midbrain. There is an area known as the substantia nigra, literally the dark stuff or the black stuff, because as neurons make the precursors of dopamine, one of the precursors is melanin, which has a dark pigment. These are important for motor control. And an easy way to see this is individuals who have disorders of motor function, such as Parkinson, might have the ability to voluntarily move, so they could move their arm this way. But as they did, there would be these tremors, because it would be a disorder affecting involuntary sections for motor control, such as these parts of the midbrain. So there is gray matter in the midbrain as well. The midbrain is very important in synthesizing dopamine and serotonin. And so these neurons which make these can then project dopamine and serotonin to other parts of the brain. So serotonin, for example, is not made in many parts of the brain. Those areas known as the RAF nuclei, which include nuclei of the midbrain, which make serotonin and then project elsewhere. The most obvious nuclei are the two bumps, the superior colliculus, or colliculi in plural, and the inferior colliculi, which then handle reflexes for vision at the superior colliculi and hearing at the inferior colliculi. And so if you're looking at sections of the midbrain, at one section you will see superior colliculi, and then inferior to that, a section would reveal the inferior colliculi. So there are important nuclei of the midbrain, and once again, important fiber tracts. I won't go through all of them. The video here goes through them in greater detail, but the touch pathways that go up to the brain start at the posterior column, but in the medulla they synapse and then form what's known as the medial lemniscus, which then travels through the rest of the brainstem, including here the midbrain. The spinothalamic tracts carrying pain and temperature information would also be traveling in this region. So there are axon tracts, and in this section, the white matter has been stained black, if that makes sense. The stain that's been used stains them black. And so here you can see the dark regions here refer to these white matter tracts. In addition, there are the tracts going down from the cerebrum, like the corticospinal tracts going down from the motor cortex to muscles, obviously have to pass through the midbrain there in orange. And we can see that here when you look at the cerebral peduncles. See how dark they are because of all of the axons traveling down here. That includes the corticospinal tract. And if you name the other ones, obviously you can see who else the midbrain is talking to, because there are connections which go back and forth between the cerebrum and other areas such as the pons. So when the names pop up in a second, very often when you just read the names of these tracts, you can determine, so the temporopontine, the parietopontine, the occipitopontine, the frontopontine, they're going to the pons, that's the pontine part, but then coming from the frontal lobe, the temporal lobe, the parietal lobe, the occipital lobe, etc. And so the midbrain has a number of important nuclei. It has a number of important fiber tracts. And one last thing is, since so many fiber tracts are going to the pons, they can then go from there to the cerebellum and form what are called cerebellar peduncles. So the cerebellum is attached to the rest of the brain. And if you look, where it's attached are often these bands of axonal tracts, this white matter here staining dark. And there are 3 cerebellar peduncles attaching the cerebellum. The superior cerebellar peduncles originate from the midbrain. So here, coming from the midbrain, are these cerebellar peduncles just inferior to the inferior colliculus, and they help to anchor the cerebellum. And so this was just a quick introduction into some of the parts of the human brainstem, and also a glimpse at their evolutionary history. The brainstem is largely conserved between vertebrates, even going back to the earliest vertebrates as we've seen.